Alright. Alright. Hello, everybody. Alright. So today, the topic is going to be what's new in Discovery Studio 2020, and I'm Adam Green. And so what is Discovery Studio? So in one sentence, Discovery Studio is small molecule and biologics discovery from scratch. Now, what do I mean by from scratch? Well, is that Discovery Studio attempts to drive early stage drug discovery from start to finish. The start is the identification part. What identification is, so you want to be able to identify a particular drug target, so like building a homology model, or if you have an experimental X-ray structure, characterizing the X-ray structure and understanding how that actually works, so that you can exploit it for rational drug design. And then once you have made that identification, then you can perform methods such as protein-protein docking or protein-ligand docking, and then examine those particular interactions. And once you know those interactions, then now you can actually start to optimize those interactions to improve the binding and the specificity. And that's really kind of the finished component for early stage drug discovery. So that's in a nutshell what Discovery Studio is used for. Now a little bit of background behind Discovery Studio. So Discovery Studio is peer-reviewed science over 3 decades. Okay, so we have implemented within Discovery Studio force field-based molecular dynamic simulations. The 2 engines that we have are CHARM and NAMD. The CHARM program was developed by the laboratory of Martin Komplex at Harvard University, and as you may be aware, this Martin Komplex was a recipient of the 2013 Nobel Prize for this work, and built on top of CHARM, separately but on top of CHARM that scales to thousands of processors and is intended for petascale class supercomputers, so you can simulate million atom systems. We have the NAMD program, which is developed by the University of Illinois Champaign. And it's important to know that BIOVIA is the commercial vendor for the CHARM program, but NAMD we provide, but NAMD is free will. And then for our homology modeling, we are the commercial vendor for the widely used academic code model. This is developed by the University of California, San Francisco, Andre Salih's lab. And then for doing protein-protein docking, we're the commercial vendor for another widely used academic code called ZDoc. This is developed by the University of Massachusetts Medical School. And we have tools that can predict protein formulation, with particular focus on antibodies that can predict aggregation propensity, viscosity, and solubility. This is developed by Bernard Trout's lab at MIT. We're also the commercial vendor for these programs. And then we have available the Catalyst database for pharmacophore modeling. And in addition, we have a robust R&D team available here at BIOVIA, where we develop our own internal algorithms. So how does Discovery Studio operate? Well, I like to think of Discovery Studio operating on 3 tiers. The first tier is you have your client interface. This is your standard graphic user interface. It can be installed on any Windows or Linux machine. And this one allows you to actually upload a protein structure, do a docking calculation, build a homology model, visualize that output and do calculations. And what the Discovery Studio client is built on top of is another one of our products called Pipeline Pilot. Pilot it has a client that's available in Windows, and Pipeline Pilot consists of modular components that can be put together into a pipeline to build custom automated workflows. So those two tiers are really the realm of what we call our computational experts. So this is where people who are computational and theoretical chemists and biologists, this is the realm that they are most comfortable with. But then we also, separately from these two tiers, we offer an additional web client that can be used by project teams and non-computational experts as well as to run calculations without having to go into Pipeline Pilot or into the Discovery Studio client, which, if you're not a computational chemist, can be a bit of a daunting task. So then of course within Discovery Studio we have a number of third-party software that's implemented. Then we have, as I mentioned, Charm, Modeler, Catalyst, NAMD, and because we're built on Pipeline Pilot, you could integrate your own third-party software to customize and personalize your research. Many of the libraries already included in Pipeline Pilot. If you have many R libraries and implemented in Pipeline Pilot 2020, we also have Python libraries such as Matplotlib and Seaborn, and Jupyter Notebooks are now available. So I talked a fair bit about Pipeline Pilot here. So what exactly really is Pipeline Pilot? So Pipeline Pilot is a graphical programming language. It consists of modular components, kind of shown here in the figure on the right, that allow you to put those components together into a pipeline. And as a result, they allow you to personalize your research with Discovery Studio, automate workflows, and then you can actually deploy those tools to project teams via web service, via like the web client that I mentioned. But I should note that if you want to use this web client, this does require an additional Pipeline Pilot server hardware. It doesn't come with a purchase of Discovery Studio by itself. So that's a little bit of the overview of what Discovery Studio is. So now for the remainder of the talk, I'm going to be talking about 4 main areas in which we have expanded our science in Discovery Studio. So the first area I'm going to talk about is rational biologics design. Now, Alex, is there any interest in antibody design or biotherapeutics here today? Okay, all right. Well, I'm going to talk about rational biologics design. All right. So with rational biologics design, a big issue is you want to be able to predict formulation properties. And it's estimated that about 90% of biologics in the market today are unsuitable for development due to low solubility issues. So if we can assess those developability issues in a very early stage, such as aggregation propensity, viscosity, solubility, then this can help us to reduce the risk of late-stage failure, minimize toxicity and immunogenicity, and extend the storage and shelf life for your antibody candidates. So how can we actually do that in Discovery Studio? Well, Discovery Studio can actually predict solubility and viscosity from sequence to structure to function in a completely completely automated manner. So we can take sequences of proteins, perform a multi-sequence alignments for your various antibody sequence, identify the appropriate templates, build homology models for your various antibody candidates, and then assess your solubility and viscosities, and then rank those so you only actually experimentally assay the candidates that are predicted to be the least viscous and the most soluble. And it's important to know that this protocol, while it was really geared towards and focused around biotherapeutics and antibodies, if you're not interested in biologics, this could be extended to proteins in general. So I just wanted to make that clear. So how do we actually predict antibody viscosity? Well, we predict it from a spatial charge map. So this is an example of the functional form. And how I think about it is, well, we're really modeling it by looking at the electrostatics of the solvent exposed residues, say in an antibody or in a protein. And with some of the test data that we have is this is from a peer-reviewed publication, and that use test data from 3 pharmaceutical companies, MedImmune, Novartis, and Pfizer. And what you see in these figures is, if you look at the red dots are the antibodies that had a very high SCM score. So the high SCM score correlates with an antibody that is highly viscous. So what this plot shows is that for each of these 3 test sets, is that with this algorithm, you're able to define thresholds for antibody candidates that are highly viscous, with antibody candidates that have a low viscosity. So then if you have a list of antibody candidates, if their SCM values is greater than a certain value, then you know you don't want to bother screening those antibodies experimentally because they're going to have formulation issues. And all of these properties are wrapped into a single protocol called Calculate Protein Formulation Properties within Discovery Studio. And you can upload multiple antibody candidates simultaneously into this one protocol. And as an example, a calculation of about 137 variable domain immunoglobulin antibody structures took about 2 hours and 27 minutes running in parallel with 2 processors on a standard Windows laptop. So this is an example of the output you would get in Discovery Studio. Here we see a table of all your antibody candidates, and you have all your formulation properties tabulated for you right there in a concise table format. And then looking at how do we actually measure the solubility of antibody candidate? Well, solubility tends to depend on hydrophobicity and electrostatics, which we can also be modeled in the same protocol. So we can look at the electrostatic repulsion, which we can model by the net charge as a function of pH. Look at the charge asymmetry, which we can model as a dipole moment as a function of pH. The hydrophobicity is modeled as the aggregation propensity, the developability indexes, and the salvation-free energy differences, which we can model as a function of pH. So your net charge, your dipole moment, aggregation propensity, developability index, and your relative salvation-free energy differences can really all be modeled within this single Calculate Protein Formulations protocol available in Discovery Studio 2020. Now these solubility predictions were trained on 21 mutations for the ribonuclease enzyme, and you see we have a good correlation to experiment of a Pearson correlation coefficient of about 0.9. Now you might be saying, well, that's only 21 mutations. That's a pretty small training set, but be aware that the publicly available data for these predictions is fairly sparse, but we've done a number of additional validation that I won't go into today on diverse proteins and enzymes that have so similar agreement with experiment. And in addition, for ribonuclease, we've actually been able to assess the pH-dependent solubility. So this is a figure from a paper where they measure the solubility as a function of various pH values, and then did an experimental fit of those solubility values for a wild type and 2 mutants. And then we were able to take that and computationally predict the solubility at each one of those pH values. So you see the dots here represent the experimental values, and then the line is our computational prediction. So our predictions were able to reproduce the experimental fit from that manuscript. And of course, this also has been shown in one of our additional validation sets has shown a similar correlation for monoclonal antibodies. So that's what's new for rational biologics design. Alright, so now we're going to switch gears and we're going to look at what's new for rational small molecule design. Alright, so our main area that we've been focusing on for rational small molecule design is in late-stage small molecule drug optimization. This can be very challenging, costly, and time-consuming. So why is this? Well, in order to optimize a particular drug, is you need to have a very good idea of the protein lead 3D structure. Ideally, you need to have some sort of co-crystal structure, maybe an NMR structure, or at least a docking post that has been informed by experimental data to some degree. Right. And then if you have that, then you need to be able to figure out, okay, well, what analogs, what modifications do I actually want to make? So you need to enumerate a whole library of those analogs, which that itself can be very challenging, tedious, and time-consuming. And then you actually have to actually make those and assay and figure out exactly which ones are you going to actually make and which ones to actually test experimentally. And we estimate that can be roughly between about $100,000 and $5,000 US and take weeks to months to actually do. And then typically we're trying to predict or experimentally estimate binding affinity of the ligand to the target, and using the binding affinity to actually measure how you're actually improving or not improving the potency, and then trying to relate that to your in vivo activity, which is not exactly a clear correlation, so that makes it very challenging. And you're making fairly small changes to the lead compound, which is very tricky to be able to do. But we want to be able to make these very small changes rather than waiting for later. We want to see if we can go ahead and make these predictions, these very small changes, much earlier on in the process, so that we can reduce our time to market. Okay. So Discovery Studio already has methods for early and late stage lead optimization. We have a variety of docking tools. We also have an MMGBSA protocol that can restore those dot poses, which is very useful for kind of an early stage lead optimization, just restoring your initial hits. And we also have a fields reperturbation protocol that is available. This fields reperturbation protocol is the NAMD fields reperturbation protocol that's been implemented in Discovery Studio. One of the limitations of fields reperturbation with NAMD is that it is currently not able to run on GPUs, which significantly slows down your performance and limits your computational efficiency. So we wanted to see, well, can we actually expand on this? And we did with a new method based on the theory of lambda dynamics. And it's for late-stage optimization. Now, before I go in and define, really talk about lambda dynamics and more specifically multi-site lambda dynamics, let's kind of backtrack a little bit and let's think about when we're doing computational lead optimization. What is that based on? Well, that's based on primarily relative binding free energy calculations. Now, how do we calculate relative binding free energy calculations? Well, we're calculating the affinity difference between two molecules. So if you look kind of to this cycle on the right, you have your ligand 1 unbound in aqueous solution, and then your ligand 1 complex to a protein, and then your ligand 2 unbound in solution, and your ligand 2 complex to the protein. And what we're doing is we're doing alchemical transformations. So recall how way back when there was a whole field called alchemy that they thought they could convert any substance into gold through alchemical transformations. What we'll do, we took that same premise and said, well, what we're going to do is we're going to chemically transform ligand 1 into ligand 2 in the unbound state, and then ligand 1 and ligand 2 in the bound state. And you estimate the frequency difference of this transformation, and then because this is wrapped around a thermodynamic cycle, the frequency difference for this transformation is equivalent to the binding free energy for the two ligands. So you get the G value. So why are we able to actually estimate frequency difference across an unphysical pathways? Well, again, if you go back to your thermodynamics, you know that free energy is a state function. In other words, all you need to know is the initial state and the final state. The path is independent. And in addition, by constructing these thermodynamic cycles, to compute relatives, we actually get an error cancellation effect, which allows for much less sampling needed to obtain reliable results. So that's a little bit of background into the theory behind relative free energy calculations. So now let's get in and talk about, well, what exactly is multi-site lambda dynamics as it is implemented in Discovery Studio 2020. Well, multi-site lambda dynamics allows you to stand multiple R groups or substituents simultaneously. So if you look in this video, you see a chemical scaffold that has kind of 3 R groups or 3 points of modification. And you look at what our chosen R groups are. We have 5 groups that we want to add onto these 3 points of modifications. So that would be 5 times 5 times 5. So that's a total of about 125 analogs that we want to be able to assess. If you want to do all this with free energy perturbation, you would have to run about 124 simulations because you're having to pair each of these analogs to your lead and run your free energy perturbation for each one to estimate your delta G value. With multi-site lambda dynamics, we're able to incorporate all 125 of these ligands into a single simulation, and thus we simulate a competitive binding assay where the substituents at each site can compete to interact with the protein. So how exactly are we able to stand multiple ligands simultaneously? Well, we take use of enhanced sampling. Particularly, this enhanced sampling algorithm is referred to as adaptive landscape flattening, and a new dynamic parameter which we refer to as lambda. So, if you're familiar with free energy perturbation at all, free energy perturbation also relies on a lambda value, but these lambda values are are in the fixed discrete windows, and then you have to figure out exactly how many lambda windows you want, and what the width actually is of each one, and how long you actually simulate each one, and this kind of requires some tinkering that is fairly system dependent. And so with this, lambda is a dynamic parameter. So when you're actually running a simulation over a defined time, each substituent is appearing and disappearing throughout the entire length of the simulation. And then as each substituent is appearing and disappearing, the algorithm is keeping track of occupancy ratio. So when a substituent is there and when it is not there. And you can think of these occupancy ratios as probabilities. You can estimate the probability of a substituent actually being present or not being present. Again, if we go back to our StatMech days, maybe we think about StatMech fondly, maybe we don't think about it fondly, but just bear with me. If we estimate these probabilities, the probability is directly related to the free energy values, so we can get the relative free energy values for each of these substituents within a single simulation. And what's important is that as these transitions are actually occurring between these various substituents, some of these substituents may be fairly large, so they may have fairly large free energy barriers that they have to overcome. So that's the importance of the enhanced sampling, this adaptive landscape flattening that actually flattens and reduces the free energy barriers and accelerates the convergence of the calculations. And this is also a distinction from free energy perturbation, because typically free energy perturbation is limited to fairly small Hall changes, but now with this adaptive landscape flattening, you could choose substituents that are fairly large and might create a fairly large voluminous cavity, which typically would be very difficult to sample with free energy perturbation. So MSRD in Discovery Studio consists of really 5 automated protocols for simultaneous relative binding free energies. The function groups that are already included in Discovery Studio 2020 include methyl groups, ethyl groups, alcohol, amino groups, methoxy groups, fluoro groups, and chloro groups. And also you can add your own custom substituents using SMILES, HOWs as well. That's available to you. And then it constructs a multi-typology system. I like to think of this as, if you're familiar with free energy perturbation in, say, NAMD, you have to construct what's called a hybrid topology, or is based on what's called a dual topology paradigm, because you're looking at pairs of ligands. Here is similar to that, except now you have a multi-topology system because you have multiple substituents that you're simulating simultaneously. And then once you have that constructed, you have to do a protocol called bias optimization. So I mentioned for this adaptive landscape flattening, to reduce those free energy barriers, those coefficients that you have to be able to estimate for each system that you simulate. So this optimization algorithm automatically calculates and estimates the appropriate coefficients for your bias potentials, so that you can lower those barriers. And then once you have those coefficients, then you can run your production trajectories and subtract out that bias and get your delta G values for all of your ligands. And unlike free energy perturbation, basic implemented in Discovery Studio is MSRD is actually run through CHOM DOMDAT on Linux for the CPU and the GPU. So maybe you're familiar with DS 2019, Discovery Studio 2019, that used CHOM OpenM on the GPU on Linux to accelerate MD simulation. simulation. Well, now with MSRD, we're actually using some DOM that on Linux for both the CPU and the GPU. Yeah, and ideally you can run MSRD on just the CPU. We don't recommend that. We recommend that you have at least one GPU and a pair of CPUs. Ideally, you have 2 or 3 GPUs available. This is a very computationally expensive task. But MSRD can be more computationally efficient than FEP. As an example, let's look at, if you look at a system that's about 49,000 atoms that has a scaffold with 3 sites and 4 substituents at each site, we're going to have about 64 ligands. If for each FEP pair, that say you want to run 32 nanoseconds, that 32 nanoseconds is a cumulative time from each lambda window times that by 64, you're going to be simulating, just over 2 microseconds of simulation. While in contrast to MSRD, you can just do a triplicate run, so 3 MSRD production trajectories of say about 30 nanoseconds apiece. So you're going to have only about 90 nanoseconds of simulation to obtain reliable delta G values for all 64 ligands. So we can say that MSRD can yield roughly equivalent results, which I'll get into in the next slide with about 20 times less sampling. And to put this into perspective, if anybody has used other free energy perturbation algorithms such as FEP+, which uses token-based licenses, you're probably familiar that free energy perturbation takes up a lot of tokens. So if you want to do a calculation like this with 64 ligands, you're going to run out of tokens pretty fast, and you have to buy more tokens. But with MSRD, if you were to, we actually offer here at BIOVIA what's called the YSU, simultaneous user complete package license, where a single user can run unlimited simulations, so you don't run into the problem of running out of tokens for your calculations, which is very important for MSRD because it is so computationally expensive. So I mentioned that it can show equivalent or even improved results to free energy perturbation. So some of the validation that we've done has been on the HSP90, which is the heat shock protein 90 system, where essentially you have kind of like 2 sites, heights and then substituents each one of those sites. And then we have data for a few of those ligands, and when you look at the actual uncertainty from FEP to MSRD, you see you have a lower uncertainty estimate for MSRD. So it has at least some minor improvement in the agreement with experiment element for MSRD. And so the last thing I want to touch on for the rational small molecule design portion of this talk is that the release of MSRD in Discovery Studio 2020 is currently a very optimistic prototype. The validations that we currently have actually tried out with Discovery Studio is still very small. The accuracy is just going to depend on the force field, and of course it's better supported on a GPU grid. They're kind of less on non-grid systems. But the reason of rolling out MSRD in 2020 is to put it in the hands of our customers. Customers have them start to use it and then give us feedback on what works, what doesn't work, so that we can improve it for future releases. And another reason why I'm particularly very optimistic and very excited about MSRD is because it's important to know that MSRD is not some new technology, some new theory that we're just coming out with in Discovery Studio. It's been developed for the past 2 decades, mostly by a single group group, the University of Michigan, Charlie Brooks's group, and they basically have come out with a number of application papers. In this one in particular, they took this particular scaffold, and they had 3 sites and 8 substituents on each one of those sites. So you had so they did 512 designs. So 512 possible designs, 512 simulations or delta G values that was embedded into a single MSOD simulation. And what they found was that of those 512 designs, that is the red dots here on the right are the relative free energy values that were equivalent to the lead compound, and the blue dots actually were analogs that were predicted to actually have a greater binding affinity compared to the lead. And then when they actually compared those results to free energy perturbation, in this case they estimated those free energies by the multi-state Bennett acceptance ratio. You see good correlation, a good linear correlation with free energy perturbation, similar to what I showed previously. And also what this same group has done, using the adaptive landscape flattening algorithm implemented in MSOD and Discovery Studio, they showed that using that algorithm actually allows you to do perturbations, to do very large perturbations. Basically, do perturbations of a 7-membered ring system, a 6-membered ring system, 5-membered ring system. Which, if you've done alchemical free energy calculations, most of those calculations are limited to very small perturbations. So you can't you can't really do scaffold hopping very well. Well, with MSOD, because we're including adaptive landscape flattening, we're able to actually estimate fairly large perturbation, or at least we're very optimistic that we'll be able to do that. And this is this is comparable with FEP Plus has a core scaffold hopping FEP plus that uses, but that doesn't actually consider the four substituent like you have with MSOD. So you see, this is the results from that paper for the 5-member, 6-member, 7-member ring, comparing that with the experimental data. So you get good correlation there. And also from this paper, because with that enhanced sampling, this particular figure shows that the number of transitions that you actually get between these various substituents with and without the adaptive landscape flattening. So with that flattening, you see the figure referred to as A. So you see lots and lots of transitions. So you can imagine if you don't have a lot of transitions, then your free energy values are likely not going to be very reliable. But you want lots of transitions like you see in A, which will lead to reliable results for large perturbations. So that's very exciting. So when I look at these results and then some of our current validation set, I get very, very excited how it is. So I'm very looking forward to seeing what our customers are going to be able to do with MSOD in Discovery Studio 2020. And so, as I said, these papers are available online. They were published in JPhys Chem Letters. Others from the Charlie Brooks's lab. So getting into our next section, we're going to talk about just classical molecular dynamic simulations and kind of improvements to that element within Discovery Studio 2020. So now within Discovery Studio 2020 is that we actually allow you to construct explicit homogeneous membrane protein systems that then be simulated with CHARM or NAMD. So you can literally take your protein, prepare it, add a membrane bilayer, and then solvate it and add your ions and then finally simulate it. So why is this important? Well, membrane proteins account for about 1/3 of the human proteome and about 60% of pharmaceutical targets. And they were able to support homogeneous in-house membrane systems such as PLPC, PLPE, DPPC. But if say you're interested in a bacterial system, like a bacterial membrane protein that exists in the inner membrane of the gram-negative bacteria, that bacterial membrane is a mixed membrane. It's about like 70-75% PLPC and about 20-25% PLPG and a little bit of cardiolipin. So how can you actually, if you have that type of system, can you still use Discovery Studio? And the answer is yes. You would have to generate that system in SOM-GUI, and then you can import that system into Discovery Studio from SOM-GUI. So that's all we need to talk about for that area. So the last area is our improvements in pharmacophore modeling, particularly with kind of two new features, interactions and ensemble pharmacophores. So in here, and the interaction pharmacophore actually replaces the previous protocol referred to as receptor-ligand pharmacophore. So before I get in and I explain what exactly interaction pharmacophore and ensemble pharmacophores are and how they work, let's just kind of again backtrack and just kind of ask the question, well, what exactly is a pharmacophore? Well, there's this definition from the paper that defines a pharmacophore as the molecular framework that carries the essential features responsible also for a drug's biological activity. Now, I think about that is if you have a series of ligands that are known to bind a particular protein, those ligands are going to interact with the proteins in a particular way and form particular interactions. Then those interactions, those ligands or those kind of common feature interactions form a pharmacophore. That's how I think about it. That's how I think about a structure-based pharmacophore. So what is this new interaction pharmacophore generation doing? Well, it's really modernizing our pharmacophore modeling in Discovery Studio. So as I mentioned before, the interaction pharmacophore replaces the receptor-ligand pharmacophore generation. Because the receptor-ligand pharmacophore generation, it only gives rid the features on the ligand, and then taste those features and see what matches with the receptor. So this was more of a ligand-centric approach. It wasn't really taking into account how the tools from structure-based drug design, because now we have docking tools and we have co-crystal structures, and we can actually look at the interactions between the receptor and the ligand. So we want to be able to build pharmacophores based on those non-bonding interactions between the receptor and the ligand, so something that's not ligand-centered. So we want to move from being ligand-centered to being more structure-based as the base, because this is the way the field is moving towards. And then so that's interaction pharmacophores. And then the second new pharmacophore features is ensemble pharmacophore generation. And the important thing about ensemble pharmacophore generation is that it allows you to generate multiple pharmacophores from very large data sets. So previously we had a feature in Discovery Studio called the common feature pharmacophore generation. but you were limited to only assessing 32 ligands for a single pharmacophore. So if you had a data set that was greater than 32 ligands, then you had to manually split that data set up into various certs, and we had some tools to help with that. Like we have a tool that allows you to find diverse molecules within that data set, so you can split that up, but you still had to kind of tinker it yourself and split it up yourself. So now with the ensemble pharmacophore generation, that's no longer an issue. So you can take a data set of a few hundred ligands, and the ensemble pharmacophore will cluster those ligands by the mass dissimilarity, and then it will automatically select 5 ligands from each of the identified clusters. So those 5 ligands would be like your cluster center and 4 neighbors. And then you'll take those 5 ligands, so like shown here, and it will do a conformational search on each one of these 5 ligands, and then estimate a pharmacophore model from the conformations of those 5 ligands, and then it'll repeat that for any additional clusters that you found. So then what you get is an ensemble of pharmacophores. So why is this useful? Well, it's useful for automation when you have very large data sets, and in particular when you have a large data set of diverse molecules that bind a protein, but they may not bind the protein all in a particular binding site, in one particular binding site. Maybe they bind multiple binding sites, or maybe they interact in multiple modes of that same binding site. So then when you want to use this as, say, a pre-filter for virtual screening, you're more likely able to select candidates that are more likely to be active when you in your experiments. So that's an overview of what's new in Discovery Studio. There's a lot more additions that I did not cover today, but the 4 components that I covered today: rational biological design, rational small molecule design, and MD simulation with explicit membrane support, and our new pharmacophore modeling features are the new major additions within Discovery Studio 2020. And with that, I'll take any questions that you may have.